Current medications improve upon this scenario quite a bit, of course. There's now an array of different antiretroviral medications that really potently suppress this viral load, up from millions, tens of thousands of copies to where we can't detect it, even when we have assays that can detect 10, 20 copies of virus per ml of blood. And it can be maintained at those low levels essentially for as long as people keep taking these daily antiretroviral medications. So obviously a massive improvement over natural infection, but one of the issues is that if anyone stops taking antiretroviral medication, the virus always comes back, usually in the course of a few weeks, and then disease will continue to progress, and those people can pass the infection on to other individuals. So this is what we want to improve upon. So even though we have these powerful medications, the rates of HIV infection around the world continue to increase over time. And partly this is because people who are infected are living longer, right? So we're not losing them from these charts, so that's a good thing. But a lot of people are still getting infected. We still have, in 2017, 2 million new infections, and we still have 1.5 million people in the world in that year dying of AIDS, even though those medications are available. So what is going on? Why is this still happening? It's complex, of course, but we like to frame this in terms of what we call the treatment cascade. There's a number of things that have to happen for someone to be people, and that's a kind of cumulative effect. So there's a lot of people who are infected with HIV who don't know they're living with the virus because they don't get tested, for example. So already this year we have only about 70% of people in the world who are infected who know. Of those people, only a subset get linked to care, have access to a physician. Most of those people get prescribed antiretroviral medicine, and then of those people, the very large majority of people who take their medications are suppressed. But if you add this up, we see that in 2017, only about half of people in the world who are infected with HIV are actually suppressed in terms of their virus, and those are the people in whom disease is not progressing. So you might think, okay, maybe this is better in the US. I don't have the stats in the US here, but the US actually doesn't do particularly well here. In the US, we have about 50 to 60% of people who are suppressed. In some countries, I was recently in Botswana, they have 80% of people suppressed. So that's kind of a story for another time. But we can do better than we are with current medications. Of course, HIV disproportionately affects sub-Saharan Africa. It's a global phenomenon, but the majority of infections are still in Eastern and Southern Africa. And I wanted to spend a little bit of time, because there's a link with the Simons Foundation here, talking about one of the issues with adherence, which is kind of an educational issue, where there's a preponderance of people who are essentially peddling fake cures of HIV. And I'm highlighting a case in Africa, which happened in the Tanzania-Kenya border, but this happens essentially everywhere in the world. And this was an individual who claimed he had a miracle cure. Take this cure, you can throw away your antiretrovirals, you don't need to take them anymore. And this is kind of the traffic jam of people going to get this cure. So I wanted to mention, before diving into the science, something that I'm involved in, which is an educational initiative put on by the International AIDS Society to try to reach out to these resource-poor settings and engage with advocates such as this individual here. She actually runs a radio program and has some reach in the community to hopefully arm them with some knowledge regarding the science and the status of the cure, to hopefully guard them against a little bit some of these false claims. And again, why I'm mentioning it is this bit of a connection with the Simons Institute, because as part of this program, we realized these advocates who are learning about cure would do well if they were empowered to go back to their community and spread this knowledge. So my partner Shai, who's a foundation here, who's an employee here, and myself made a modest donation, which was matched by the Simons Foundation Employee Matching Program, and we were able to leverage that to get a donation from Chevron and put together an actual grants program to be awarded in 2019 and to continue. So I wanted to just acknowledge and thank the Simons Foundation for that. So those are fake cures of HIV. There are also real cures of HIV. There's at least 2 of them. So some of you may have heard of this gentleman who's an outstanding advocate and all-around good person, Timothy Ray Brown, also known as the Berlin patient, the first person to be cured of HIV. More recently, he has some company in someone called the London patient. This was, front page news in the New York Times of a second individual has been cured. So how did this happen? How can we cure 2 people? Is this scalable? Unfortunately, the scalability of this is going to be a real challenge. There's no easy way around this. The way these people were cured, they unfortunately had cancer. So the first person had leukemia, the second one had Hodgkin's lymphoma, and they had to have a bone marrow transplant. So their immune system is ablated by chemotherapy. And there's a mutation called CCR5 delta 32, which is known to exist in about 1% of Caucasians. Unfortunately, it's only in Caucasians, really, that makes cells not able to be infected with HIV because they don't have the receptor. So when these people had bone marrow transplants, the donor cells were from people who had this mutation. So they received an immune system that's resistant to HIV. And in Timothy Ray Brown, the virus didn't come back for 10 years, and the London patient were 2 years out, and the virus hasn't come back. So we think it's possible to cure HIV, but this process is very risky. There's more than double-digit mortality rate. It's not something we want to give to people who have controlled HIV infection. So that's all I'm going to say about the transplant scenario. Now I want to switch to something that I think will hopefully be more scalable, which is trying to harness the immune system to try to cure HIV. So if we want to know how we might leverage the immune system to control HIV, the first thing we might want to do is learn from nature and look at different outcomes that occur in natural infection. So I told you about kind of the natural course of HIV that happens in most individuals. About 1% of individuals are something that we call elite controllers. These individuals are able to control HIV to undetectable levels without antiretroviral therapy. Their CD4 counts are stable, and they do really pretty well. So what is special and different about these individuals? So to address this, a group that was pulled together by Bruce Walker, and this benefited also from some philanthropy, pulled together a group of 974 of these rare individuals. This was a really global effort, and had a control group of people who were progressing, and did a genome-wide association study to see which polymorphisms were associated with this protection. And you can see one really strong hit here, and nothing else reaching above this threshold. So what is this hit that is genetically associated with elite control? It turns out this maps to something called MHC1, Major Histocompatibility Complex Type 1. And this gives us really a very clear idea of at least part of the way that elite control is working. Because the job of MHC1 is to actually show virus infection, to show essentially what's being produced in a cell, to an arm of the immune system called CD8 T cells, which can kill these infected cells. So the virus comes in, the viral proteins start being produced, this is presented on MHC1, the CD8 T cell sees it and kills it. So since that genetic hit is associated with elite control, it really makes a pretty strong case, like an extremely strong case, the CD8 T cells are important in this process. So let's move and talk a little bit more about cytotoxic T cells and what we know about them in HIV infection. And this is our protagonist, CD8 T cell, cytotoxic T cell, for the rest of the story. We normally see them as little pellets in the lab, so this is actually a little bit frightening, but I'm glad they're usually on our side. And here they are ganging up on and essentially working at killing a target cell. So let's zoom out just a little bit from this, and I'll show you some movies I took. These are CD8 T cells in red, moving around under a microscope in a collagen matrix. And what they're doing is they're scouring around looking for, in this case, infected cells, because their job is to find and kill these infected cells. So this is happening in people's bodies all the time for various antigens. It's kind of always fun to see into this little hidden world. So what happens when they see an actual infected cell? In red, again, we have a cytotoxic T cell. Here's a target cell. It's going to see it. It's going to kill it. You can see the cytoplasm of the cell leaking out. And then it stays in contact with it for a while. Here comes a second cell that's also going to get recognized and killed. So we call that serial killing. The third target here gets away. So they're called cytotoxic T cells. They're sometimes called killer T cells. They're named because that's exactly what they do. They have other functions, of course, but we'll focus on that killing function. So cytotoxic T cells are important in controlling HIV infection, but it's not as simple, of course, that only people who control have cytotoxic T cell responses to HIV. Everyone has cytotoxic T cell responses to HIV. And actually, the first person to find these T cell responses is Professor Nixon, who's in the audience here. So these responses emerge early in infection, and another clue that they're important in controlling HIV is that as these CD8 T cell responses come up, that's when we see the viral load come down to set point. And that happens to everyone to some extent. Elite controllers do it better and control to a lower level and more durably. So what's special about the T cells in these elite controllers? So we know it's associated with MHC1, and different of these alleles, this is highly polymorphic in the human population, and different alleles show different parts of the virus. So maybe it's a matter of T cells that target the virus at certain spots are more effective than others. And this turns out to at least be a good part of the truth. So this was a study, again, by the Walker Lab, where they mapped a lot of T cell responses, asked what part of the virus are being targeted, and then they built this into a model to predict whether or not someone would be a controller or not. And you can see this is just with MHC type alone, which we know is associated with control. If you actually then build into the model the parts of the virus that are being targeted, the model does better. And actually, all we need to know is the parts of the virus that are targeted for this to perform very well. We can actually leave out HLA alone. So it's not still the full story, but an important part of the story is that T cells that target HIV in vulnerable spots tend to do better. Why might this be? Well, HIV, one of the big challenges is that it really rapidly mutates to escape from any pressures that we put against it. And this is true of antiretrovirals. We know that from the days of AZT. Monotherapy, HIV escapes very quickly. It also mutates to escape from T cell responses. So this is an epitope. So this is a sequence of amino acids that the T cell happens to recognize. And this is a study just showing all the different ways that this particular epitope can change that allows it to escape from HIV. And this is actually a target that's pulsed with a escape peptide. We can see there's some interaction, but it doesn't form a stable synapse. So these cells either don't recognize the infected cell at all, or they recognize it sometimes less well. So maybe the inability to escape is an important part of this elite control. And I think this is true. Again, it's probably not the full story. And this really highlights that this is still a very active area of research. This paper came out a few weeks ago. And what they did was look at the amino acid sequence of HIV in a different way. They brought in structural information and built a network model with this. So essentially, they brought together a lot of information regarding amino acid to amino acid interactions that any particular site might have, and then gave that a network score. And what they found is that these amino acids that had a lot of interactions tended to be located in these vulnerable parts of the virus, the parts that were targeted by people who controlled infection, probably because they're more constrained, they're less able to mutate, so the virus kind of gets trapped more easily than something that targets these amino acids out here, which are relatively isolated. So I bring this up. This is not exactly my area of specialty, but there's, I think, some interesting math involved as well, and I know that that's an area of interest for the audience, so people may want to look some more into this. So the idea is, if we can really figure this out, if we can really target HIV where we want to target, can we back it into a corner, essentially, and achieve elite control in the majority of people? And that, I think, is an open question and an open challenge. So the virus can escape by changing itself, but that's not the whole story. You find responses in people that target parts of the virus that aren't escaped, and nonetheless, the person is progressing to AIDS sometimes. Nothing's really absolute in human biology. So another part of the story is that the T cell response undergoes something that we call exhaustion. And essentially, what this is the T cells fight HIV or any chronic infection for a long time. The immune system essentially realizes it's not winning. It's probably doing more damage because these cytotoxic T cells are releasing molecules that can be toxic. So there's a regulatory mechanism by which it starts to shut itself down. And it does this in part by expressing what we call exhaustion markers, LAG3, TIM3, PD1, and others. And the Nobel Prize in Medicine this year went to James Allison, who was initially working with CTLA-4 as well as a Japanese group. So this is also important for HIV, and I want to pause here for just a second to make a point on how different areas of research cross-fertilize each other. So these checkpoint blockades, they're called. These are ways of blocking these exhaustion pathways, have really been revolutionizing cancer treatment in recent years. So anti-PD1, for example. And actually, the first demonstrations that these are functional in the human immune system came from studies of HIV. So there's plenty of counterexamples as well, as you'll see later, where discoveries in cancer inform HIV and, of course, other areas of human science. It's all the same immune system working in these different contexts. Okay, so that's all great, but here we suppress HIV to the levels where we can barely detect it. We still have these CD8 T cells. The virus can't mutate anymore. How come the CD8 T cell can't finish the job and eliminate the infection in this context? So I'm going to go over some of what's known and some of the dogma in this field and paradigms, and then introduce some of our work, which I think pushes this in some different directions. So one of the reasons that some T cells might not be able to eliminate the last of the HIV is that that virus might have escape mutations. We talked about that before. So maybe if we can redirect the immune response against non-escaped epitopes, we can start to clear out those infected cells. Maybe the CD8 T cells are exhausted. If we use some of these therapies that are used in cancer, like anti-PD1, maybe we can reinvigorate it. One thing I didn't talk about is that the infected infected cells can compartmentalize themselves at anatomical sites that CD8 T cells don't go to very often, such as certain areas of lymph nodes called B cell follicles. Maybe we can redirect T cells into these sites. And finally, HIV can go into what we call a latent state, where it hides from the immune system, and we might have to wake it back up if we want to kill infected cells. So I think the last barrier is the one that I would say receives the most attention, so let's spend a bit more time talking about this. So we saw that HIV is a retrovirus, so it inserts its DNA into the host cell genome. After that point, it's an inseparable part of that cell. There's no way of getting rid of that provirus without killing that cell unless you evoke some different technologies like CRISPR and such, but typically it's there for good. It can persist for years or decades, the immune system can't see it, it can reactivate and then produce virus and restart infection. So the dominant paradigm, I'd say, in the HIV cure field is that if we want to eliminate the reservoir, we have to do something called kick and kill it, or shock and kill. It's the same thing. So you start with one of these latently infected cells, it's hiding from the immune system. You treat it with something called a latency reversing agent, so you wake it up, it shows itself to the immune system, and then you have one of these cytotoxic T cells that I showed you earlier coming in and doing what they do best and killing these infected cells. So this approach has made it to the clinic in a number of different trials. Unfortunately, the results so far have been disappointing, and this is just kind of a typical summary from one of these trials. No change in the reservoir with kick and kill. Maybe they didn't wake up the virus strongly enough, the latency reversing agent was too weak, maybe the epitopes are escaped. It's not exactly clear why this didn't work. So why do people think that this kick and kill approach will work in the first place? A lot of the evidence supporting it comes from what we call latency models. And how these latency models work is you take a CD4 cell, activate it, infect it with HIV, and then let it go back to a resting state in vitro, all in the test tube. And then you say, this looks like the latent reservoir. And if you do that in vitro, you can shock and kill these reservoirs, and that works really fine. What our lab really focused on is trying to do the same kick and kill combination against CD4 cells from people living with HIV, so taken directly out of the body. It sounds like really an obvious thing to do. The challenge is that these infected cells are present only around 1 in a million, so you really need a lot of cells to be able to test this. So we get around this using a process called leukapheresis, and we're able to test this. Okay, so when we try to bridge this latency model where we're able to eliminate the reservoir, and a clinical trial where we can't eliminate the reservoir by trying to eliminate it in patient samples, what we find is that when we treat with a latency reversing agent to wake up the virus, we see maybe a trend towards increasing the reservoir, and when we do this kick and kill combination, we don't see any decrease in the reservoir. So this was a surprise to us. We actually thought we were just crossing T's and dotting I's. We thought this would work, and it's led us down really a different path of exploration, which is what we're going to talk about really for the rest of the talk. And this was published in 2018, and we made the argument that these latent reservoirs not only are hiding from the immune system, but are resistant in some way that we don't understand. So when the CD8 T cell tries to kill them, it may not work as we expect. And one of the pieces of evidence from this study is that even though we couldn't kill these real infected cells from the patient, we could reactivate that virus, infect new cells, and those same CTL, killer T cells, could kill those new cells. So it really seemed to point to something intrinsic about this initial infected cell itself. Okay, so that then leads us to the work that we're doing in our lab to try to see how these different cells that actually harbor HIV might be intrinsically resistant to being killed, even if they're recognized by these cytotoxic T cells. And another fun movie for this. So this is the CTL in red. Here's a target cell in blue. And the point that I just want to make is there's a long interaction between the two of these. There's a lot of two-way communication. If the target cell decides to die, it's an apoptotic process, so it's a programmed death. It essentially kills itself. There's a lot of things that could happen on the target cell side that could influence whether or not that cell dies. There's been a lot of study directed on the effector side. There's been less study directed on the target cell side. So we decided to go after this question, and this is the experimental setup that we use to get at this. We're going to look at what we call real condition and a mock condition. Both of these cases we have CD4 cells, which are the targets of HIV, and they're 50-50 labeled with a green dye or a red dye. In the real condition, half of these cells are also labeled with HIV to make them targets for the killer T cell, cytotoxic T cell. Here there is no HIV, so there's nothing that's actually a target. We then add these cytotoxic T cells to try to kill the targets. Here we see some are killed, a subset survive. Here nothing happens. You'll see why that's important in a second. And then we sort these by flow cytometry based on the color. So we get these survivors, these cells that should have been killed but weren't, these bystanders, and these might be affected by the fact that these CD8 T cells are recognizing HIV, because these produce a lot of cytokines and chemokines, a lot of danger signals that change the transcriptional profile in surrounding cells. And in this mock situation, there's no reason that these two should be any different from each other. So what that then looks like if we take this data, so I should mention, we then profile these with RNA-seq, which is a deep sequencing method that looks at all the RNA that's being produced. And then this can be simplified onto a principal component analysis plot, which just essentially clusters conditions that are similar to each other versus those that are different. So here we have what we call the mock conditions, the ones that were labeled with the two colors, but remember they weren't different from each other at all. And the difference between these and the survivors and the bystanders is not what we're really interested in. This is just the environment that these cells were in, because these activated T cells produce all these danger signals that change the transcription. What we're really interested in is the difference between these two populations. They were in the same environment. These ones weren't targets. These ones were targets, but they survived anyways. So we, of course, can't study the ones that were killed because they're gone, but we can infer something by the depletion that happens here. So this is RNA-seq data, which is plotted in a way that just lets us look across the genome at different cellular processes. And the more gold something is here, the higher the significance is, for the difference between the survivors and the bystanders for what was depleted. The first thing that came up was cell cycle. So we looked a bit more closely at this. And this is a representation of the RNA-seq data. So the more green that you see in some plot here, this is the proportion of genes in that process that were differentially regulated between the two conditions. So the point here to see is just that there's a really comprehensive coverage of this pathway. So what we saw is that cells that weren't dividing preferentially survived. So this was known from some time ago. It wasn't known to me, and I'm not sure it wasn't exactly known to the HIV field, but it's been described previously that cells that are not in cell cycle are resistant to being killed. So far this is just essentially a nice confirmation of things that were available. What's more rich here is the data set of what was actually enriched in the survivors. So the previous was depleted, and now this is enrichment. And we see the immune system lighting up. So of course, this is something we're interested in. And if we zoom in on this, we see that the pathways that are differentially regulated are involved in this process of antigen presentation that we talked about earlier, this way of getting the virus processed and presented on the cell surface so it can be recognized by the CD8 T cells. So again, no real surprises, but nice confirmation that the method worked. So where things got a bit surprising is looking at this pathway of programmed cell death. And we saw an interesting hit here, which was overexpression of a protein called BCL2 in the cells that survived this killing. So BCL2 is generally a master regulator of cell survival. It is involved in the decision of the cell to die or survive in response to all kinds of stresses and insults. It's not usually thought of in association with CD8 T cell killing through granzyme perforin, but it is a role has been implicated by some studies. So this is how T cell normally kills. It punches a hole in the protein of the target cell by something called perforin, and this allows this protein called granzyme to come into the cell. And granzyme is typically thought of as cleaving caspase 3 here, which sets off a cascade, which results in the cell dying. But it can also act by cleaving this protein BID into TBID, which causes death through the mitochondrial pathway. And BCL2 can inhibit this by sequestering TBID. So there is some reason to think that this BCL2 could be involved. So with this hit, we decided to do some functional assays and see if we could validate this. This is a similar experiment to what I showed before. The target cells are pulsed with peptide. We try to kill them, and then we just directly assess the level of the survival protein, BCL2, in the cells that survive this killing process. And you can see that the more peptide we add, so as we go across here, we have more cytotoxic T cell killing, the higher the levels of expression of BCL2 in the cells that survive. And we can look at this at a cell number level also. We see we're getting killing depletion of the cells that have low levels of BCL2, but not those that have high levels of BCL2. So BCL2 seems to be able to protect the cells from dying, but what does this have to do with the HIV reservoir, so those cells that survive for a long time in people on treatment? So remember, these are present at a frequency of 1 in a million, so they're really hard to study, but we were able to adapt a way to actually directly phenotype these cells from the blood, and that's what's shown here. These are probably 10 million CD4 cells from the blood, and here's the few HIV-infected cells that we pick up. And we can look at expression of BCL2 in these cells. This patient isn't our most extreme case. I think it's this one here. So there's some variability in individuals, but what we see is that we always see higher levels of BCL2 expression in the HIV-infected cells versus the uninfected cells. Is that just a consequence of being infected with HIV, or is that something unique about this long-lived reservoir in these individuals? So to ask this question, we did the same thing in cells from people who aren't on therapy. So here the virus is replicating. There's a lot of cells that have been newly infected. And if we do the same analysis, we don't see any differential expression. So there's more work to be done here. We need longitudinal studies, but what we think the data is pointing us towards is some kind of selective process where the cells that live for a long time in these people have higher expression of this pro-survival protein, and that may be involved in why we have a hard time killing them, both in vitro and in these clinical trials. Okay, so again, there's this interplay between cancer and HIV, and here we can borrow something from the cancer field that might be helpful for us. So BCL2 is also involved in survival of many types of tumor, and there's a BCL2 antagonist, which is actually a licensed drug called venetoclax, which is used to treat chronic lymphocytic leukemia. So this again just binds to an active site and inactivates this BCL2. So we thought, given this new insight, can we take this BCL2 antagonist, and is this maybe helpful for trying to eliminate the HIV persistent reservoir? Before getting into combining it with a cytotoxic T cell, we had to do a bunch of background work and see whether or not this drug alone was enough to deplete the HIV reservoir. This is a busy slide. There's no need to understand everything here. It's also a ton of work by Yen-Chin Ren. The take-home message is this drug alone does not reduce the HIV reservoir in these ex vivo patient samples. We didn't necessarily think it would anyways, because where we think we're going to have the activity is combining it with these cytotoxic T cells. So we went forward and tested these new combinations, and this is, I think, pretty novel work. So this is looking at total HIV DNA. This is looking at the infectious virus using a viral outgrowth assay, where we require the virus to replicate in order to measure it. And what you can see is where we have this triple combination of the cytotoxic T cell, the latency reversing agent, the BCL2 antagonist. This combination is where we start to see significant reductions in the reservoir, and these are maximum likelihood estimates with 95% confidence intervals. So that's just a few cases. We've of course done this with a number of individuals. Here each line represents a different combination of target cells and cytotoxic T cells. These are the double combinations where we don't see a consistent effect. And here's the triple combination where previously we weren't able to reduce the reservoir with just the CTL and latency reversing agent. Now we can see some consistent reductions in infected cells in vitro. So we think that this process of going to unbiased RNA-seq has given us a hit that we're able to follow up on, at least in vitro. And since this is a approved drug in CLL, perhaps it's something that can be tested in the clinic, even though the side effect profile is a bit complicated. So that's for the clinicians to sort out. Okay, so in the last part of this talk, I'm going to talk about whether these sites where HIV integrates in the genome might have some effect on whether or not we can kill those cells with the cytotoxic T cell. And this part's a bit preliminary. It has a question mark at the end. It's work in process, but it's something I think we're interested in, so I thought I'd share it today as well.
Before you go to the next part,
please do.
What is other than T cells do that could cause effects?
Right, so BCL2 is always in attention with pro-death molecules such as BAD and BAX. So this tension between the two determines whether or not a cell undergoes apoptosis through the mitochondria. So we do see non-specific cell death with this BCL2 antagonist that we have to control for in order to see if there's a specific death of the HIV-infected cells. So what we hope essentially is that by mining the data with the RNA-seq that we showed earlier, hopefully there's other and more specific and more targeted pathways that we can tip this balance as well. But at least this is kind of one way where we could finally get reductions in the reservoir. Any other questions on that part? Okay, So the next question is, does it matter where this HIV integrates in terms of whether or not that cell can be killed by a cytotoxic T cell? And this is something which the field has thought of in terms of whether this integrates in an oncogene, something that affects the survival or proliferation of the cells. But what I think is unique about our work is we're asking whether it influences whether that cell can be killed by the immune system. So why would we think that the integration site of this virus might have an effect on the function? This is known to happen at the level of organisms. So this is the difference essentially between an orange and a blood orange. It's not HIV, but it's a retrotransposon, so it has a lot of similarities with the cycle of HIV. It doesn't have an extracellular phase, but it copies and pastes itself in the genome. And just by inserting the retrotransposon at this site, we have this clear phenotypic difference. Another example, especially for wine connoisseurs, is actually the difference between Cabernet and Chardonnay is just an insertion of this LTR retroelement and the promoters at this site. We can go one step further. If this recombines and gets rid of the retroelement part in the middle, and we only have the promoter left, now we have another varietal of grape. So at an organism level, the movement of these elements can have a phenotypic difference. So the question we're asking is, can this also have a functional difference at the level of an individual cell? And to do this, we used a similar method that we used for the RNA-seq. We infected cells with HIV. These are flow cytometry data. The cells in this window are infected. We tried to kill them with a cytotoxic T cell. We see we kill most of them, 63%, but there are some that are still left. So let's characterize